So now, assume that these are our samples from each of the people here. Okay. Over here we have Mrs. Clean. We're going to go Mrs. Mr. Baby, Mrs. Mr. Baby, Mrs. Mr. Baby. Okay, so the cleans, Mrs. Clean, Mr. Clean, Lane Clean, Mrs. Pupil, Mr. Pupil, Rose Pupil, Mrs. Deceased, Mr. Deceased, Sarah Deceased. Then over here, this is sample A, sample B, sample C, sample D. So I'm going to load these samples in order into the electrophoresis gel. Okay, and we'll show you loading them, but you need to remember that order so that when you interpret the gel, you know exactly what each person has as far as the samples and the colors. Okay. All right, now I'm going to fill the wells of the gel, starting from the far side to the front. Since we have 13 wells, every single well needs to be filled. So I'll go in here. And fill the first well. Change the pipetter tip, so that was Mrs. Clean. Fill the second well. And that would be Mr. Clean. And the third well being Lane Clean. And I'll turn this off and continue. Okay, and so here's our last well. Sample D. All right, now all the wells are filled. And we'll go ahead and hook up the electrodes to the lids. Again, we want to run to red. And we will turn on the unit so that we run to red. Okay. So now I'll run this for a few minutes, then we'll look at the results. Okay, so the gel is running. It's been about 5 minutes. We can see those bands moving up the gel with the white pattern in the background. We can start seeing those separate. Right, heterozygous individuals will have 2 bands. Homozygous individuals will have 1 band. And we can start seeing a lot of bands on those individual people. I'm going to change the angle so we can get a better look and you can see the separation going on. So here's the gel and we can kind of see what's going on. So over here we're starting with Mrs. Clean, Mr. Clean, Lane Clean, Mrs. Pupil, Mr. Pupil, Rose Pupil, Mrs. Deceased, Mr. Deceased, Sarah Deceased, sample A, sample B, sample C, sample D. Okay, we can see, I can see 4 different colors here. We can see one color here is yellow, and we see a purple, we see a red, we see a yellow and a purple, yellow and a blue. So basically we have a yellow, a red, a blue, and a purple. Okay. Now tough ones, let's see here. Tough ones then. This one right here is a tough one because there's a purple band. So this is purple and red, but let's look here. So it looks like a purple and red. Okay. This one is a yellow, or sorry, this is a purple and a yellow. This one's a yellow and a red, yellow purple, yellow blue. This one's a purple and a red, which is tough. This one is a red and a blue. Okay. Then it looks like a yellow and a blue, red and yellow. This one looks like a red and a blue, purple, yellow, red, yellow, blue, yellow, red, yellow. Now that we have our gel ready, the next step is interpreting the gel. Typically, if this was DNA, there would be no colors. So instead of the colors, you'd have just a simple band. That band would move a certain distance, and all we would know about the DNA is its relative size. So that's what I want you to look at here, is basically view this as these colors as bands. Okay, so we want to convert these bands to actual allele or sizes. All right. So to convert these colors to sizes, we're going to use table 10.1 from the lab manual. So here's table 10.1. You can see that we have 4 markers represented: TPOX, CSF1PO, DS5818, and THO1. So those are all the markers we're going to use in this study, right? Typical FBI have 13 markers. Paternity tests have 16 markers. We're going to use 4 so that you don't have to do too many. So the colors represent TPOX. So what we ran out then was TPOX. And if you look at TPOX here, you can see that we have alleles. We can see STRs, short tandem repeats. We have a short tandem repeat of 8. We have a short tandem repeat of 9, of 10, of 11, of 12. If we have bands on the gel, then each of these short tandem repeats is represented by a single color. So on the gel, then, we have the colors yellow, we have red, then we have purple. One color that's not on the gel is a green, which occurs between purple and blue. So on the gel, it would go red or yellow, red, purple, green, blue. Your next task then is to convert the colors on the gel to STR lengths. Okay, for example, let's say that Mrs. Clean is purple and yellow. By being purple and yellow, then the purple would correspond to one of the STR lengths in TPOX, and the yellow would correspond to another one. Okay, yellow being the smallest, then the yellow would correspond to an STR length of 8, and the purple would correspond to an STR length of 10. Therefore, STR lengths for Mrs. Clean would be 8 and 10. So finally, your assignment in the lab report associated with this gel is to take the people and the colors on the gel and convert them into STR lengths using table 10.1 for TPOX.